So now we will talk about the cellularization of Drosophila embryo and how the cellularization will lead to the formation of a cellular blastoderm. So here the formation of the cellular blastoderm will happen after the migration of the nuclei from the center to the periphery. Once at the periphery, okay, so by the cycle 14, these nuclei will start to be surrounded, each of them by its own cellular membrane. So how this process of cellularization occur? So here if we take a section from the peripheral part of an embryo, you see here we have the nuclei at the periphery, okay, and the cellularization will start by membrane invagination to the inside. You see this is the membrane, and we will have an invagination to the inside to form kind of canals called furrow canal. Okay, so these kind of furrow canal will separate the different nuclei from each others, and these invaginations, they come from the cell membrane. So by this step of cellularization, we notice that the size of the nuclei is much bigger. So we can have a nuclear elongation. Okay, the nuclei will be much more elongated than before. And this is due to the secretion or the synthesis of one protein called Gugelkern. So Gugelkern protein is produced, and this will elongate the nucleus or the nuclei, all of them. So the first step of cellularization is mediated by the microtubules. Okay, so this is the first part, let's say the apical part, while the basal part of the membrane will be formed due to the actin complex. Okay, so first the microtubule will be required, and then the actin, which is another cytoskeleton element, and this is important for the formation of the basal membrane. So you see that this process, okay, is mediated first by the microtubules, and when they tried, for example, experimentally to add the nocadazole, okay, during this cellularization of Drosophila embryo, they added some drugs, okay, one drug called nocadazole, and they have seen that the cellularization process is completely stopped or inhibited by this drug, since nocadazole blocked the activity of the microtubules. And this will confirmed at that time that microtubule activity is important for the cellularization process, since when we block the microtubules by nocadazole, we block the cellularization. So by the cycle 14, by the end of the cycle 14 in Drosophila, the embryo will be formed by 6,000 cells, okay, present at the periphery, and this stage is reached 3 hours after fertilization. So during the cleavage, okay, during embryogenesis, we have nuclear cleavage or nuclear divisions, and you see that during the nuclear division we have the nuclei at the center. Okay, each nucleus is surrounded by a clear, you see this zone, a clear zone, okay, highly rich in microtubules and microfilaments, so in cytoskeleton elements. Why the nucleus at the center is highly surrounded by microtubules and microfilaments? So they have found that these cytoskeleton elements around the nuclei are essential for the migration of these nuclei from the center to the periphery by the stage 9, 10, 11, and 13. Okay, because from the cycle 9 to 13 we have the syncytial blastoderm stage, and we have the migration of these nuclei from the center to the periphery. So we call the nucleus and its surrounding micro cytoskeleton, let's say. So these nuclei and their associated cytoplasmic islands. It means these accumulation of cytoskeleton that make kind of island around the nucleus. So these are called energets. Okay, so energets are the nuclei and their associated islands formed by microtubules and microfilaments. So during the embryonic nuclear cleavage in Drosophila, 3 hours after fertilization, we mentioned that we get the cellular blastoderm stage. Before the cellular blastoderm stage, we have a syncytial blastoderm stage. So this cellular blastoderm stage represents the cycle 14. Okay, this cycle 14 is also called MBT, and this MBT stands for Midblastula transition stage. So the Midblastula transition stage, it's a critical stage during embryogenesis, since everything before is called pre-MBT and everything after is called post-MBT. So it's a very critical and important stage during development. So why the MBT or the cycle 14 or the cellular blastoderm stage is highly important? So if you look at this graph, we can see the modification in terms of messenger RNAs, okay, during time. So from the fertilization, passing through the blastula or blastoderm stage, in Drosophila it's called blastoderm, and then towards the gastrula stage. So you see that by the cycle 14 or the cellular blastoderm stage right here, we have a transition from maternal gene expression to zygotic gene expression. This means that we'll have a decrease in the messenger RNAs of the maternal genes. What are the maternal genes? The maternal genes are genes expressed by the mother, and they are inherited by the embryo at the very early stages. Okay, so it means here in this figure, so this stage, this one, and this one, we have maternal gene expression. So these genes are expressed at the level of messenger RNAs, and they are inherited from the mother. By the Midblastula transition, by the MBT stage, these maternal genes will be decreasing. Okay, we have a drop off of this expression, and we'll have the expression of new genes called zygotic genes, and these genes are specific for the zygote. So it means for the embryo from this stage until the different other stages of development. So the Midblastula transition, as we mentioned, it's a stage of transition, okay, from maternal gene expression towards new zygotic gene expression. So I mentioned last time that by the cycle 11, so during the syncytial blastoderm stage, we have very small amount of zygotic genes will start to be expressed. So it means that a bit before the MBT stage, okay, but by the MBT stage, the main zygotic genes will start their expression. Okay, so by the MBT, we start having a slowdown in the nuclear division. So we will have a slowdown in the nuclear division, because the aim now is to cellularize these nuclei, okay, and not to form more nuclei by mitosis, okay, or by nuclear divisions. So we have a slowing in the nuclear division at the periphery. We have a cellularization, okay, so we have a slowdown in the nuclear division, we have a slowdown in the cellularization as well, and we have an increase in the new RNA, in the new zygotic RNA transcription. Okay, so here slowdown nuclear division and cellularization, and will have new zygotic gene expression. So the maternal genes will be degraded, and the new genes called zygotic genes will start their expression. So the question now is how the maternal genes will be degraded by the MBT stage. So here they found out that we have an RNA binding protein. It's a protein that will bind to those maternal messenger RNA in order to degrade them. And this protein is called Smog. They took the name just from one movie that came out at the same time of their study. This movie was The Lord of the Rings. I think you heard about this movie. It was very famous movie at that time. So, and in this movie, Smaug was the dragon that will destroy everything. That's why they called the messenger RNA binding protein, so this protein that will bind to the maternal messenger RNAs, Smog. Okay, just to make the story a bit funny, because I mentioned that in Drosophila they always call the genes according to the mutant phenotype. So here Smog will destroy all the messenger RNAs during the MBT. So what is Smog? As I mentioned, it's an RNA binding protein that will bind to specific messenger RNAs in order to destruct or to degrade them. So they tried to mutate Smog. Okay, they made a maternal Smog mutations. Okay, in Drosophila. So we got embryos without any Smog. So it means without the protein that will degrade the maternal messenger RNA. So this embryos, okay, showed a slowing down in the nuclear division. Okay, they don't show, a slowing down in the nuclear division. So we have a disruption in the slowing down of the nuclear division. Okay, and also this prevent the cellularization. We cannot get a cellularized blastoderm. And also they found that the maternal mutation of Smog prevented as well the increase in the zygotic gene transcription. It means it will prevent this transition from the maternal genes towards the zygotic gene. So it will disrupt the transcription of the zygotic genes. So what is Smog? So Smog, it's a protein. Okay, it's translated from a Smog maternal messenger RNA. So this gene is also maternal gene. It's expressed in early stages of Drosophila embryogenesis, but the highest levels, so the peaks of Smog expression, okay, are the highest, okay, by the MBT stage. Before the MBT, we have low levels of Smog expression. Why? The highest levels are by the MBT stage, because it's needed at the MBT in order to degrade the maternal messenger RNAs and to induce the expression of the zygotic genes. So Smog is very essential for the progression from maternal genes to zygotic or nuclear development.